So the final talk is going to be a double-headed contribution. The first head is is Paul Emsley, long-standing developer of the popular molecular graphics package Coot. But I think it's going to end with a fancy demonstration of virtual reality.
That's the plan. Oh, hello. I'm going to go to displays. Is that what I want to do? Arrangement, unmirror, click, function F5. Good. Right, indeed. Thank you for the invitation. Let's see how we get on. I have 60 slides to do in 10 minutes. It seems. Okay, so the Coot is a program typically used for crystallography historically. Now it's being used for Cryo-EM. In such, the way in which you use the tool is different. So for Cryo-EM model building, at least the way I use it, we typically need to move more atoms than we do when we're editing crystallographic models. The maps are routinely lower resolution, and the starting model is further away from where we would want them to be, and typically systematically so rather than just randomly. This has been, addressing these needs has been the focus of my attention for the last couple of years. Not every part of what I've been doing is visible at the GUI, and we'll talk about that. I will talk about 4 things, perhaps less now. Jiggling, morphing, restraining, and refining. These things have been improved as a result of conversations with my colleagues. Oh, G&P, thank you for reminding me. If you aren't using G&P routinely and you're doing your model building, then let me encourage you to learn about the key bindings and give them a go. I find I'm using G&P very frequently for navigation, to move around the structure. Okay, so for what resolution is Coot useful? It's where you can see the side chains really, and if you can tell the difference between a purine and a pyrimidine, that's a sort of a resolution at which Coot is more comfortable. You can try to use Coot at lower resolutions. It's more of a struggle. More prior information needs to be used in the refinement and model building. Of course, I understand that's an issue. There are problems that to be solved at that resolution. And it's a topic of ongoing research. So my eyes were opened to the needs of Cryo-EM by my colleagues, conversations with my colleagues Alan Brown and particularly Alexei Muntz, who collected high-resolution mitochondrial ribosomal subunits. We were encouraged to help them out by editing our software to, changing our software to help them do the model building of the ribosomal subunits. We wrote a paper on that. It seems to be quite well cited in the field now. That's nice. And additionally, and subsequently, I had the opportunity to work on this structure you'll hear about more later on from Laurie Passmore, and the first author here is Annika Samuel. Okay, but moving on. Oh yes, indeed. There's also, I was approached by Abe and Jonathan Grimes from Oxford Struby, and they had a structure where they were trying to model the genome packing inside of a coronavirus and said, what can you do to help us here? And I said, that's an interesting problem. And I was unable to help them at the time, but now I think the Coot features are now there to solve that problem that they had 8 months ago. They did it with Rosetta. So the tools that are available and are being modified are Spherifine, Spherifine Plus, where you're using more residues. You can use regularization. There's a thing called tandem refine, where you are refining along the structure rather than in a sphere. I'll be talking about how one can use Gemin-McClure distance restraints or modifications of distance restraints by Gemin-McClure estimators. And we'll see about the other things. So there are a number of structures in the mitochondrial subunits, the red and yellow here, that either needed to be modified by mutations or built de novo. And there are some tools in Coot that were added. One of those I'm going to briefly mention is jiggling and morphing. Just a couple of slides on each. I don't have so much time to elaborate. The idea is that we have a structure, a model. In this case, it's a piece of RNA, so it's easy to see where it should go. In the general case, it's rather less clear where a model should go. You are somewhere close to where you want to be, within a few angstrom. It does a rotation translation search and places it in the density. So it is a fast molecular placement feature where you are approximately where you want to be, or you have put the molecule approximately where you want it to go. You're asking the question, how am I rotating and translating the atoms? It's a 6-dimensional problem. The translation problem is local, though. It was originally designed to work on ligands, but when I heard my colleagues discussing the problem, I thought, I have looked at that problem before, and instead of moving 10 atoms, let's try to move 400 residues. It doesn't work quite so quickly. Okay, so very typically, you might operate on a chain instead of a residue. So this is the operation. It takes a few seconds, and part of my work is to make this work using all of the processes on your computer. So let's take in some re-engineering inside of Coot, which you can't see, but it just goes faster as a result. So that was the representation of the backbone. Here's an all-atom model. You can see it doesn't quite fit the density, and what we would like to do is make some systematic changes to the model so that it fits the density, but the changes at each residue position, or nucleotide position in this case, are related to its neighbors, so that it doesn't get too distorted. So that's what I was just saying there. So for each residue in the chain, this is a description of model morphing. We ask, where does this one want to go, and how can we combine that with where its neighbors want to go? So we, for a particular residue, we do a residue selection, up and downstream to residues, and ask, where does a fragment that looks like this want to go if we were to refine it in the map? And we can see, we get out of that a rotation translation operator. We ask that question for each residue in the structure, and then combine the rotation translation operators with averaging and its robust system, so we are rejecting outliers in the rotation translations, and combining them and applying each of those rotation translations in a local environment to bring the residues closer to the high density, high density parts of the map. Okay, I don't have time to talk about that. You can read about that. I put the PDF online if you're interested. This is what it looks like in operation, something like this in a few steps. We are moving to a solution that puts the atoms closer to the middle of the density, and if I turn on the real structure now, you can see that after a few cycles, it takes about 5 or 10 seconds each cycle, you get a clear answer that's pretty close. This was applied both to proteins and RNA, in the case of the ribosome here. We started the structure that's labeled A, and after a few cycles of morphing, then we got to solution 3, morph 3 here. You can see this. The idea of what I'm trying to illustrate in this point is that the final model is not so different from the one we got after a few rounds of morphing. Okay, so that's morphing done. Now we'll talk about refinement, and the way refinement has been improved is by using prior knowledge. So the tools for providing the prior knowledge were being developed in the lab already by Garib and his student and now postdoc Rob Nichols. The program is called ProSMART. The idea being that we can use template distance information to provide stabilization in our refinements. So, for example, we've solved a molecular replacement problem at low resolution, and this is the APO form. Last year, we have solved the structure, which is the holo form containing the carbohydrate. It has the ligand bound, and not unsurprisingly, this diffracts to a high resolution. What we want to do is take the information from the high resolution structure in a local fashion and apply that prior knowledge in terms, expressed in terms of local distances to stabilize the refinement at the lower resolution for the model using the low resolution data. It looks something like this. So what I'm showing you here is that the distances were taken from the green structure and applied to the brown structure, and then in Coot they are colored. Where it's gray, these two distances are the same. Where it's red, they're further apart, and in blue, closer together. And what we'd like to do is, what we're trying to express with this particular figure is that around the ligand binding site, we have additional information where we know that it's quite clear to us that we don't expect conservation of backbone distances around the ligand binding site upon ligand binding. So those distances are coloured red, and then we can go into Coot and edit them and delete them. And say we don't want to apply these distance restraints in our low resolution model. We can write these out and then use them in Coot and RefMac for refinement. The target, so thank you. So very typically, one would use sidechains as well. So this is illustrating with just a backbone trace. One uses sidechains as well to have additional stabilization information routinely these days. Okay, so the target function is modified from the typical least squares or harmonic restraints that Coot uses pretty much everywhere else in its refinement. So now we're using a Gemmen-McClure modifier of this target function so that when we get a long way away, for some value of a long way away, it's user adjustable. Then we no longer pay a penalty, not so big a penalty, between hypotheses that are a long way away from the target values. So if you were having a least squares competition between a model that was this distance apart and this distance apart, then there would be an enormous penalty for the structure that was further displaced from the ideal value. We're not paying that penalty anymore, so we can have conserved or consistent deviations of distance with this weighting scheme. Additionally, apart from distances, we can have other geometric information that we can apply to our model, our prior knowledge. And in this case, we're looking at the output of LIBG, which does base pair stacking and parallel plane restraints, which can be used in RefMac and Coot for stabilizing and refinement. LIBG provides Watson-Crick base pair stabilization, and in this case, wobble and reverse wobble. If you look at it in Coot, it looks something like this. The sticks don't do this representation justice. When you can see them in 3D, it's a lot more useful representation, and you can see, because they're rotating, the ways in which certain bases are not well aligned or not parallel in this case. So there's packing between, so this is an example of LIBG restraints in operation. You can see that one of this base is twisted, the purpley pink structure, before refinement, and then when the refinement has been applied, you can see that the restraints have made it parallel, and they are, that's the parallel. These are now paralleled, and there is also base stacking interactions that are consistent with our prior knowledge also. And we can do interactions between base-base and base protein, for example. Here we have a nucleotide here and a phenylalanine, so that when this restraint gets turned on, you can see that even though it's inconsistent with the density, it's no longer like this, it's turned over the phenylalanine to have aromatic stacking interaction. Okay, so previously, Coot had a naive or high resolution, they're the same thing these days, interpretation of how to do model building of rotamers. And what it did was it cut the description of, it cut up the atoms that were being fitted into residues. So it made a cut along the peptide bonds. And it said, I'm going to fit these, but not these. Now, of course, that turns out to be naive. You can't see my pointer? Ah! Thank you. What is that? I've never had that before. Can I see it? No, I can't. Okay, so Coot is cutting the bonds at C and N and C and N. And only these atoms are being modified by the algorithm. At least that's how it used to be. And moving on, what we're going to do is take a set of high probability chi-angles. They're high probability because they're low energy, and try to add those on to a C alpha, C beta model. Now, we can't just edit the chi-angles because that won't find the correct solution in our model. We need to have another set of hypothesis generation, as well as merely the chi-angles. So previously, the way I used to find other hypotheses is allowing rigid body refinement. And what that meant was that in low resolution maps, the atoms got sucked down into the density and it destroyed the main chain or damaged it quite badly. We obviously don't want to do that, so we're using the formalism from the Richardsons to use the previous and next residues to construct a so-called backrub vector around which we are rotating atoms. The trick being, or the point being, that the carbonyl, oxygen, and the nitrogen of the residues to which the central residue is attached are also moved as part of the optimization, which wasn't the case before. Okay, and then we can apply rotations around this vector and add addition of our chi-angles, and we will get a set of hypotheses which do a reasonable job at finding real solutions in mediocre resolution maps. Okay, so then we can score each of our rotamers according to fit density. This is a comparison. This is how it used to be, falling down. New method, starting from the same place. You can see I've turned on backrub rotamers. If you are unfamiliar with the buttons here, click right mouse in this bar and a whole new world of Coot interface will be open to you. Okay, so when we apply the method, then we get a different solution to the one we've got previously. This one doesn't fit the density so well, but the main chain geometry is still well preserved and it doesn't try to crash the atoms. Comparison of the two structures, you can see the one that we like is sticking out of density. Ho-hum. So if we look at a typical X-ray map and compare it to a Cryo-EM map, we know that it's even when the phases are good, we expect for good structures for the atoms to be sticking out of density. So it's okay. It's not great, but it's okay that we have atoms sticking out of density. Right. I'm conscious of time running out. There is a new module for building carbohydrates, so that would have been fun to look at your maps there with your rescaled maps. Maybe we could have done that yesterday. So that was published last week. Thank you, Charles. I hopefully have made it useful for Cryo-EM maps. Of course, the proof will be in the pudding. I have reworked the interface to show map properties in a way that you may be familiar from other programs. We have interactive markup of Rotamer goodness with polyhedra and also Ramachandran goodness by coloured balls. Also, there is the interactive dots that you may be familiar from model probity. But these are interactive. As the model is refining, these dots are getting updated because the program is now all multi-threaded. Well, all multi-threaded, my goodness, that would be something of an overstatement, but in parts it is. To activate these things, you set show intermediate atoms, Raman markup 0. That means I've turned it off. Okay, put 1 there, put 1 to turn it on. turn it on. Okay, we can find some things that are new for Cryo-EM in the extensions modules Cryo-EM. It's got some new tools in it, including this one, which was, what's the word? Suggested by Ian. So there's a tool for sharpening and blurring. Very typically, I don't like, very typically, the output of a Reliant, or those to which I have been exposed by my colleagues, it's kind of notchy and ugly looking, particularly in Coot. So routinely using RefMax to sharpen, blur, and resample it upon Coot, and they look much more elegant and comfortable to build in rather than this notchy thing that you get from the EMDB. Right. Oh, also, there is the kurtosis-based B-factor optimization. I don't know how it works for Cryo-EM. You are welcome to try. Additionally, we have the Ramachandran plot, which you can have selections for now, so that if you wanted just the A chain or 200 residues of the A chain, you can concentrate on those rather than the whole thing. Hooray, thank you, Bernard Locamp. Right, okay, I am going to finish. I have left Hamish some time, and I will take questions after Hamish has finished, if that's okay. So thank you to my colleagues at the LMB, my collaborators, Kevin and Bernard, and those who provided the dictionaries. Thank you. Okay. Right, so what... Oh, I've gone off. Ha, hello, again. So what we're going to do now is have Hamish speak about how to use the preferences so that I can show you the displays. And after that... Okay, so he'll talk about how to set up the interaction of Coot VR.
Okay, thank you very much, Paul. Great. So what I'm presenting here, I'm afraid to say no flashy demo. I do have these, but they're props. I'm not actually going to be using them. What I've got here is essentially just what it is useful for you to know if you're going to see the demonstration that's behind that wall over there. Okay. It's just outside. All right. So, Coot VR. Totally serious. It's Coot in VR. It's not a toy. It is not outreach. The suggestion is, the hope is that all of the functionality of Coot can be put into VR. This is the idea, and so if there's something that you can do in Coot, I want you to be able to do it in Coot VR, and that's what I am working on. Why would you want to do this? What is the impact of working in VR? Well, first to think about it in a bit of an analogy, so I've got two pictures here, one of knitting and one of the inside of a cockpit of a plane. Why have I got these two things? Well, if you imagine a person who's knitting, they use their hands and they look at it directly in front of them, they interact with their knitting in 3 dimensions. If you were to say to a self-respecting knitter, how about, I've made a simulation in which you can knit on a computer screen with your computer mouse, nobody would be interested in doing that. And because they know that there's a lot of fine motor skill and 3-dimensional intuition that you get from having something in 3 dimensions in front of you. And the cockpit of the plane, I'll get to that in a second. So to be clear, it is, with Coot VR, I would claim at least, it is easier, i.e. faster, to specify intended destinations in 3 dimensions for atoms during refinement. Right? Sounds pretty useful. Like faster as in, I don't know what the factor of it is, but if you can save a bit of time on that, then it's a good thing. Easier and faster to notice features in 3D and to navigate your molecule. It is significantly better than 3D monitors. I suppose a lot of you will have those 3D monitors. Everybody who tries out VR knows that they are better than those. Number 3, you can navigate menus faster due to increased screen space. And I'm using these because there's no such thing as screen space in virtual reality. You'll see what I mean. And that's the cockpit thing. I mean, imagine taking all of those controls and squashing them onto a screen that sits in front of you like this. Like a pilot wouldn't like that very much. And in virtual reality, I can pretty much put anything anywhere. I have a lot of space to work with when designing interface. And finally, you can make it easier to train people to use Coot. Possibly this will mean that a BSc student can produce a highly refined model for a final year project, and they are less likely to procrastinate on Facebook, because it is impossible to tab away to something else when you're in virtual reality. Then a clarification about this. If you are an experienced Coot user and you are skeptical of what I just said, I would especially like for you to try Coot VR and give me your thoughts. If you think that Coot VR is cool or awesome, that's really lovely. It's lovely. People always have such a nice reaction to it. But I am interested in a specific question, which is virtual reality useful to model building or useful to structural biologists? So I am interested in whether you consider Coot VR to be a good value proposition and something that you would actually use in your life, in your work. Okay, so some controls. This is the thing. I've got it here. You're going to have this in your hand. Essentially, there's only 3 buttons I'm making use of. There's a trigger button at the top, which you press with your index finger. This is your index finger. This is your middle finger, and I'm facing my palm towards you, so it's not rude. So index finger does one button, and middle finger does the other button. And there's the thumb button on the front of the thing. The analog stick can also change the contour level. Let me know if you would like to change the contour level. It's a bit sort of risky at the moment. I'm still working on it. Okay, common mistakes, number 1. Forgetting glasses. It's fine to leave your glasses on inside the headset. You know, I should have maybe brought some glasses onto stage, but you can have them inside here, right? But just be a little bit careful so they don't get caught. Put the headset on back first, so don't go like this. Think of it like ski goggles or like water goggles. Go like that. Okay. It's nice and stretchy like this, and you've got these. You've got Velcro straps to strap it onto your face. It is, you want it snug, right? Not suffocating you, but you want it. There's a location that your eyes are expected to be at, and you want to put them there. Otherwise, it'll be blurry. Yeah, use the straps. You've got to adjust it on your face. One mistake that people make is they take this controller and they hold it like that, which is a little bit like holding a mouse like this. You don't hold something by its buttons. You want to be conscious of whether you are pressing the buttons. Okay, common mistakes number 2. Not putting your hand all the way over to the thing that you want to grab. This sounds pretty ridiculous, but this is quite common. Let's say that I want to pick up that phone. Something that happens is that people are like, okay, why am I not grabbing the phone? And it's like, because you need to move your hand over to this. And I said, you need to move your hand over to it. And someone once said, no, I don't, it's right there. It's three-dimensional, this world that you are in, okay? You want to move around and look around. You don't sit there stock still. You don't want to move too much at the same time, so don't go under the desk or anything like this. This isn't so much of a problem. This certainly hasn't happened today. And finally, wire anxiety. So lots of people get very paranoid that they're going to trip over the wire, or that they're going to throttle themselves with the wire or something like this. This never happens at all. I've never, in 3 years of working on virtual reality, I've never had any problems with the wire. Frequently asked questions. Number 1, how much does the hardware cost? £300 to 400, basically. Any electronics retailer. How much more time am I going to work on this project? About at least 1 year. Do I plan to turn this into a crowdsourcing, gamification, or citizen science project? No, I don't. Is the human race doomed because people will spend all day in virtual reality? Also, no. Are you going to make it so that I can see how molecules behave when I punch or squeeze them? That's molecular dynamics, and that's not what I'm working on. So almost certainly I'm not going to do anything like that unless I was working on this project for 10 years or something like that. it's not clear that it would be useful for model refinement. Am I going to add voice control? Possibly I will add voice control, but only for accessing molecules by saying their serial number. I'm not going to make it so that you just say to the, I'm not from Star Trek. I'm not making the computer that you can just ask to do things that you want them to do. And can you use this for outreach? All I can say is that's not its intended usage. You can take a picture of that, and if you really want some VR outreach, that is something that I made. But yeah, that's not what this is for, promise. Feedback form, it would be great if you could fill this out. Have you ever used normal Coot? Have you ever refined a whole structure with normal Coot? I'm just trying to gauge your level of experience. How sick did you feel on a scale of 0 to 10? What would you most like to have added to Coot VR? Please give some thought to this. Would you want to use Coot VR for refining structures at your office once it is fully implemented? And I've got 4 refining structures bolded there because some people would want to use Coot VR at their office to entertain their kids, but that's not what I'm interested in. If not, why not? Why would you not be interested in using Coot VR? Okay, and finally, nausea, vomiting, and VR sickness. Or motion sickness. This is a problem that I've never had. My software has never made anybody sick, apart from me, because I test on myself. It's very understandable for people to be concerned about it, because you might have tried other kinds of virtual reality that weren't so good. We know we know the tricks now, is what I'd say. I'm not going to go into detail. But with that said, if you feel queasy at all, immediately take it off, even a little bit queasy. That's not happened, but yeah. If you have any detailed thoughts or comments, please send them. There's my email address, and also I need an examiner for my PhD, so if anybody's interested in that. Okay, let's stop it there.
Yeah, will do. Okay, great, thanks. I have to say I tried it this morning, and it didn't make me sick, and it didn't hurt me in any way, so I would highly recommend having a go. So any questions for either Hamish or Paul? If anyone's worried about coffee, we're going to extend the coffee break, so there's no rush.
Should have clarified. No, it doesn't.
What's the question?
Oh, sorry. The question was, does it already have all the features that are in... Does Coot VR have all the features of Coot? Not right now.
But you can build a protein model.
Can you build a protein? Yeah, you can. Yeah. You totally could. It would be really... It would not be better than using Coot right now, but you could do it.
Okay, any other questions? Okay, so before we thank all the speakers, we're running about 15 minutes late, so we can be 15 minutes late back from coffee, and I think we have plenty of time after the last session before the dinner. So if we could just thank all of this session's speakers, thank you.